This video is on Chapter 3, Genetics, which reproduces the variation that we see amongst life. Some quick facts before we begin Chapter 3. First off, there are several YouTube videos posted within these slides. They are all linked on the slides that are posted on D2L, as well as a separate page in D2L under the supplementary resources for this chapter. Please do take the time to review them at your leisure. In other words, as you are able, as you have time. They do go into more details than we have time to get into in this class, but if you're feeling rusty on any of your high school biology, these videos are a good supplement for that. Don't forget to rewatch as many times as you need. That's part of the wonderful format of having things online, is that you can take the time to rewatch as many times as it takes. Also, exam number 1 will be open from this Friday, June 19th, through Sunday, June 28th. There's a study guide that'll be posted on D2L. It's also linked in these slides. There'll be a separate video going over that and how to prepare for these exams. Keep in mind that there is a 2-hour time limit on exams. However, that is a suggested, not an enforced time limit. So if you are in the exam and you go over that 2 hours, it will not kick you out. It will not automatically submit. It will not give you a 0. It just is a timer for your reference, and so that I know whether or not the exams are about as long as they are supposed to be. They're not supposed to take you something like 5 or 6 hours. That said, if it does take you 5 or 6 hours because something happens, you have to go let the dogs out and they run away, you have to go to the store, kid needs attention, whatever the case may be, I will not be docking points for that. Just send an email if there's any difficulties, or if you the internet goes out and you have to navigate back to the page and it won't let you back in or anything like that. Just let me know and we'll troubleshoot those on a case-by-case basis. But again, 2-hour time limit, that's suggested, not enforced. So this chapter looks more in depth at DNA. Last chapter talked about it, and we talked about how it was discovered by Watson and Crick, and really Franklin, Watson, and Crick. But DNA is a small molecule that's kind of like a code. It tells the cell what to make, how to make it, and thus how to make a living organism. DNA is packaged into chromosomes. So the genetic code is on these chromosomes, and that tells the cell what to do, that tells the cell how to do things. So you need to make this protein because you're a muscle cell, or you need to do this because you're a heart cell that's conducting an electrical impulse. Ultimately, this code is the basis for most human variation that we see, although not all human variation, which we'll see in Chapter 4. So to start off, we need to talk about what a cell is. Cells are the basic unit of life. There is nothing that we consider alive that is smaller than a cell. There are biological materials, things like prions and viruses, subcellular machinery, but we don't consider those to have all of the characteristics of a living thing. But your body is made up of trillions of cells. Of those trillions of cells, by number, most of them are prokaryotic cells. They're not actually you. They are instead your endosymbionts that live in your gut, bacteria that live on your skin, live pretty much anywhere on your body. There are bacteria. That's not to say that they are necessarily bad, because they give you a lot of ability to resist disease. They prevent the actual bad bacteria from taking hold, oftentimes. In the case of your endosymbionts in your gut, they actually help you digest food. You couldn't digest food without them. So looking at the structure of a prokaryotic cell, if you watch that video, you'll remember that these cells, they are much simpler than eukaryotic cells, or than the cells that make up the human body. They do have cytoplasm, and in that cytoplasm, there are cellular machinery, there are molecules of DNA, there's a lot of stuff going on, but it's not separated at all. It kind of all is just hodgepodged together. Everything's just kind of in there somewhere doing what it needs to. Prokaryotes are the oldest kinds of organism. We have definitive evidence of them as old as 3.5 billion years, but we have circumstantial evidence, or not direct evidence. We can't zoom in with a microscope on a rock and see cells from before that time, but we have evidence that they may have existed as old as 4.1 to 4.2 billion years. So prokaryotes are very old. They're also very simple. They are single-celled bacteria. They don't have a nucleus, so their DNA is free to just kind of go anywhere in their cell. They don't have double membrane bound organelles, which just means they don't have a nucleus, they don't have mitochondria. They have organelle-like structures, but they don't have organelles or the subcellular machinery that we see in eukaryotes. An example of a eukaryote is you, so animal cells. This video takes you through what the differences between a bacteria and a animal cell are. Their animals are just one of several kinds of eukaryotic organisms. There are protists, which are actually a bunch of different kingdoms, fungi, plants, and animals. We obviously are animals, and one of the things that signifies that we are an animal and not a plant is we don't have a cell wall, and we have mitochondria without having chloroplasts. Again, all of that is covered in this YouTube video, as well as the associated videos on Crash Course Biology's channel. So looking at the eukaryotic cell, these are much more recent in evolutionary terms, 1.2 billion years old-ish. That's when we have the first direct evidence of eukaryotic cells. So cells that have a nucleus, they have mitochondria, and if they're a plant, they have chloroplasts. Some eukaryotes are single-celled. All multicellular organisms, including humans, are eukaryotic. There are no multicellular protists. Protists are single-celled only. Eukaryotes can be single-celled or they can be multicellular. The defining trait of a eukaryote, U means true, so EU means true, and karyote actually means kernel or corn. So it comes from the fact that all eukaryotic organisms have a true nucleus. They have that true little area where the DNA is contained within. That does several functions. It helps protect the DNA from pathogens. It helps protect the DNA from any kind of damage from outside of the cell. And notably, eukaryotic cells have organelles. Specifically, they have those mitochondria and or chloroplasts, depending on whether they're a plant or an animal. Within the human body, so thinking about the human body and the human body's cells, we have all these different kinds. Every human body cell is a eukaryotic cell. But of those eukaryotic cells, we have somatic cells and gametes. Somatic cells are basically every cell in the body. These cells have a full complement of DNA. So in humans, that's 26 chromosomes. In many of the other ape species, that's 48 chromosomes. Humans, 46. These cells reproduce through mitosis. Mitosis can be very long and involved. There is a video here to help guide you through that, as well as the lab for talking about the different phases of mitosis. For our purposes, we really are concerned with what is the end result and how does it get there. So it goes through several cell phases: prophase, metaphase, anaphase, telophase, and then you have two cells that are identical, or at least mostly identical, to the parent cell. The other kind of cell here, gametes. There's a video here on meiosis, which is the process of taking a somatic cell, a normal body cell, and turning it into a gamete or a sex cell. The gametes, at least in most eukaryotes, are the egg or ova and the sperm. Each of those, the egg cells and the sperm cells, have half of the normal amount of DNA. So in humans, instead of having 46, they only have 23 chromosomes. So they only have half the normal amount of DNA. They reproduce via meiosis, so it's the process of taking a normal body cell with 46 chromosomes and breaking it down into 4 cells with 23 chromosomes each. I know that math doesn't sound quite right, because if you have 46 to begin with and you break it in half to 23, should you only have 2? Well, the process involves several steps that are similar to mitosis, but in meiosis, the cell essentially goes through mitosis twice without replicating the chromosomes the second time. There are lots of details of this process that we are not going to get into in this lecture, but they are there in the lab and in this video for your viewing pleasure. So thinking about chromosomes and the form DNA takes in these eukaryotic cells, most of them are bound in chromosomes. DNA is packaged in these chromosomes, at least for eukaryotes and for the kinds of organisms we're worried about in this class. The chromosome number varies by species. You can see here many examples of chromosome numbers by species. Keep in mind that organisms with fewer chromosomes are not necessarily less complex or less evolved, quote unquote. Instead, the chromosome number really just helps function to tell a sex cell whether or not it can combine with another sex cell. Because many eukaryotic organisms send their reproductive cells out into the environment rather than targeting a single individual. Something like coral, for instance. Coral is a living animal, and it reproduces by just sending its sperm and egg cells out into the ocean and hoping that they eventually come into contact with another coral's reproductive cells. Thus, this number of chromosomes helps to ensure that they are reproducing with the correct organism, the correct kind of coral, and not some other random organism in the ocean. So when we look at DNA, DNA is the molecule that transmits genes. These genes are found on chromosomes, and all of these chromosomes together give us the genome of a organism or of a species. Nuclear DNA is I'm going to mess this up, homoplasmic. So homoplasmic, meaning that there's really only one kind, homo, one, and it is consistent throughout. So it is one kind of DNA that's bound into these chromosomes that is sent through the reproductive life cycle. Other organisms, things like bacteria, often will have multi-form or multi-modes of DNA, and so they reproduce in a very different manner. The kind of DNA that would reproduce like that in humans is mitochondrial DNA. So you have your DNA. If you send your DNA into 23andMe or to Ancestry.com, they sequence it. That is your nuclear DNA. Your mitochondrial DNA is a little different. It is not found in the chromosome in the nucleus of your cells. It's found conveniently in the mitochondria. So mitochondrial DNA in the mitochondria. Who would have thought? But this mitochondrial DNA, because it is not the same structure as nuclear DNA, it's not in these chromosomes, it is much easier for that DNA to accumulate changes over time. And those changes allow us to determine haplogroups, which is really just a fancy way of saying it lets us figure out what person ultimately gave rise to another. So it lets you trace your matriline, your mother's 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 mother, back through generations. The structure of DNA, if you recall from the lab, there is a sugar, phosphate, and a base, and it's bonded down both sides. So the two sides are the sugar phosphate. In this case, deoxyribose is the kind of sugar. So nucleic acid, deoxyribonucleic acid, it's that deoxyribose sugar around the outside twisting around. The base inside of that, those kind of rungs of the ladder, or those colored segments there in that image, those are what really carry the genetic information. There are four kinds. Oh, sugar, phosphate, and a base. Adenine, thymine, guanine, and cytosine. We abbreviate those as A, T, C, and G, respectively. These are complementary bases, meaning that A always pairs with T and C always pairs with G, and vice versa. T always pairs with A, G always pairs with C. These complementary bases let us figure out things like what is the complement for CAAAAT, or as I like to say, cat. It would be ktsa. So C pairs with G, A pairs with T, A pairs with T, A pairs with T, T pairs with A. So the two sides of that DNA strand are mirror images, mirror in terms of A to G, A to T, and G to C. There are some things to keep in mind, though. These do not always pair up correctly. In a perfect world, A would always pair with T, C always pairs with G, but there is error introduced. The errors are very rare in terms of how often this DNA is replicated, but it does happen. And that can be what we call a SNP, a single nucleotide polymorphism, SNP. Those, when we see those, we can track the origin of those. They happen randomly, but often with very little effect overall. And so we can track who's related to whom that way as well. So we can follow mitochondrial DNA to figure out matrilines. And by tracking SNPs, short or single nucleotide polymorphisms, that allows us to trace non-maternal or non-mitochondrial DNA back through generations. When we look at the structure of DNA, it does have to unzip in order to replicate. So as the DNA is sitting there, certain enzymes come in, specifically DNA helicase, which unzips the DNA and pulls it into these two separate strands. What we call them the leading strand and the lagging strand. The leading strand is easier for the enzymes to replicate, so it can kind of just latch on and go brrr all the way down. On the other hand, the lagging strand, basically the lagging strand is facing the wrong direction. And so the enzymes can come in, attach, but they cannot just read in one direction. They can in attach and have to read the wrong way. So they come in, attach, make new DNA, detach, come back in, lower down, build until the point where they kind of already had built stuff, come back out. And there are biochemical reasons for that. It's not just because it's cool. It's because the structure of the sugar molecules make it impossible for the DNA to be read the wrong direction. So it can only be read in one direction, and so because they're the two strands, one can be done all the way through, the other one has to be done in segments. Before we get into the details of mitosis and meiosis, we need to think about what chromosomes are. Chromosomes are structured in homologous pairs. Homo meaning same, so homologous pairs are the same kind of chromosome. You get one from each parent. That's part of the whole point of having the number of chromosomes during meiosis. So you get these pairs. In humans, we have 23 pairs for a total of 46. There are 22 pairs of autosomes in humans, and autosomes are not sex chromosomes. So they're the chromosomes that make up most of the human body. 22 pairs of those. We have one pair of sex chromosomes. That is your X and Y chromosomes. In a biologically typical female, she will have two X chromosomes. In a biologically typical male, there will be an X chromosome and a Y chromosome. They are named X and Y not because of the actual structure, but because of the size difference. If you look at an X chromosome, it is much, much larger than a tiny little Y chromosome, which we'll see examples of here in a little bit. Because a male has both an X and a Y chromosome, there's a 50-50 chance that each of the male sperm cells will have an X or a Y. And so really it's the male that determines the sex of the child, at least in a strictly genetic biological sense. And that is done by either the presence of the Y or the presence of the X in the sex cells in his gametes. Again, that is typical, and these are broad strokes here. There's a lot of nuance that can come into play. Just said both of those. Good job, Ian. Good job. So looking at a karyotype takes that karyote from eukaryote, so that nucleus, that little kernel, and makes it in a form that we can see. It actually involves, this is actually how it's done, taking a photo of the nucleus, cutting out each chromosome, and pasting them together. That's how it's actually done, at least in times when we don't have high-powered gene sequencing. And this is not Thursday, September 12th, but there are those two videos linked in this slide for you. So, thinking about what's going on with a single fertilized cell. You started off as a zygote, just like all other humans on Earth. You started off as a fertilized egg cell. So the two half chromosome complement cells, the 23 and 23, came together to make a 46 chromosome cell. That's the diploid number, the number you need, generally speaking. Your 23 chromosome sex cells, your 23 chromosome gametes, are haploid. They have half the normal number of chromosomes. But during this time, your body is going through mitosis at an incredible rate. So from the moment of fertilization through ultimately to the moment of death, your body is constantly going through mitosis, the process of making new cells. So in this figure, you can see each of those phases, which are interphase, prophase, metaphase, anaphase, telophase, ending with cytokinesis and the separation of the cells. For our purposes, it's okay if you just know that during interphase, it's the phase in the interim, i.e. the boring phase. The cell's doing what the cell does. It's not until prophase, metaphase, anaphase, and telophase that we're really seeing mitosis occur. In meiosis, so not mitosis, but meiosis, I didn't name them. If I had, I would have given them very different names so that we could keep them more organized in our heads. But in meiosis, an egg cell is produced or a sperm cell is produced. But if it were to just go through the normal mitosis route, it would end up with double the number of chromosomes, 92, which is not what we want. It does not end up creating a viable offspring. Instead, the number of chromosomes has to be reduced. So it replicates and reduces during meiosis. Go from one progenitor cell or one original cell to 4 offspring cells that are going to then go and combine with other gametes to make the next generation. There are things that can happen here during this process. It can happen during mitosis, but it's more rare. Meiosis is more common because it's more complex. There's more failure points. There's more things that can go wrong. And one of the things that can happen is a translocation, where part of a chromosome is pulled away into the wrong daughter cell. So some of the cells are lacking that part of the chromosome, and some have too many. We can also see non-disjunction. Non-disjunction refers to a failure of the chromosomes to separate. And so one of these 4 gamete cells will have a double copy. That can result in things like Down syndrome, which is trisomy 21, trisomy meaning 3, and so trisomy 21, 3 copies of the 21st chromosome because of non-disjunction, because those 2 copies didn't separate correctly during meiosis. So in humans, keep in mind that haploid cells have 23 chromosomes. There are no pairs, just 23 chromosomes. And that recombination can occur. Recombination is kind of the whole point of meiosis. Recombination allows for genes to be shuffled even more than just as chromosomes. So it allows sections of chromosomes to kind of overlap for a second, and when they pull back apart, they're sticky. So they pulled part of each other off and swapped those genes between each other. It's what we call crossing over. This can result in haplotypes, clusters of genes that we generally see occurring together. And because when that crossing over happens, it's not just one point, it is the whole kind of chromosome touching. And so generally they'll get pulled together, resulting in these haplotypes, resulting in general linkage between these different genes. Translocations can also occur along with non-disjunction. Translocation refers to the full separation of just a segment of chromosome. So oftentimes it'll be one chromosome kind of gets broken and then pulled into another chromosome. So instead of being the normal length, it ends up as kind of double length chromosome. So obviously we've been talking about DNA and chromosomes, but this is exactly how we see it play out when we reference the previous biology we've talked about. Mendel's law of independent assortment, so saying that genes are carried independently of each other and have an equal chance of going into an offspring, that arises because of these chromosomes and because of the way this plays out in molecular biology and statistics. So each of the genes that Mendel looked at were on different chromosomes in the pea plant. It's very fortunate because it allows for us to see that independent assortment. Each of those chromosomes has an equal chance of going into the offspring cell, a 50-50 chance, which can give us these nice big Punnett squares. That doesn't work if the genes are located on the same chromosome, which is part of why there has been some debate over whether or not Mendel was miraculously lucky or just collected a bunch of data and kind of figured out what looked the best when put together. This takes you through a more in-depth example, which you're free to read through in your book or through the slide, but this video is already getting quite long, and I want to keep things moving. So when we look at DNA and protein synthesis, DNA ultimately doesn't really do that much on its own. It's the fact that it builds proteins. That's what we need it to do in the human body. There's 7 types of proteins listed here: enzymes, structural, gas transport, antibodies, hormones, mechanical, and nutrients. All of those together come from needs that the body has. The body needs enzymes to do certain things. It needs structural proteins to make a body to begin with. You need your hormones to regulate cell function and cell metabolism. All of that is controlled by the DNA and certain segments of that DNA. So how does DNA do this? How does DNA make proteins? Ultimately, proteins are made of amino acids. So amino acids are the building blocks of proteins. There are 20 different kinds, and in human, 12 of them the human body can manufacture, no problem. So they are not considered essential. The other 8, however, can only be obtained through food. So you have to have them in your diet, and that's why we call them essential amino acids. Each of these takes various combinations of the genetic code to say, hey, we need to make this. And that's done through initially transcription. Transcription refers to the process by which we create messenger RNA, and that messenger RNA will then go and tell the rest of the cell how to build our protein. Transcription, think of it like you're writing a transcript, right? You're taking the spoken word and writing it down as a visual representation of your spoken word. So we're taking the DNA and transcribing it into messenger RNA, mRNA, inside the nucleus. So you're taking one form of nucleic acid and going into a different form of nucleic acid. Translation, on the other hand, translation occurs when we take the mRNA code and go into an amino acid sequence or into the protein at the ribosome. And at that ribosome, the codons, or the 3-unit code of the genetic material, will be read, and the correct amino acids will be pulled and chained together, linked together. That process is translation. So transcription goes from like the spoken word to the written word, or from DNA to messenger RNA. Translation would be like taking the spoken word and putting it into a new language, putting it into Japanese or Spanish. So you're taking it from one form and putting it into a completely different form with totally different rules and totally different functioning. That is the process of going from from nucleic acid sequence to amino acid sequence, or going from nucleic acids to proteins. Just like during DNA replication, during transcription, we have to unwind the DNA so we can read it. If you can't read the DNA, then it doesn't let you actually use it. As that DNA is unwound, certain regions are selected by cellular machinery, and that'll go in and say, okay, this is where I need to start reading from. And it'll just read that section of the DNA. That section gets replicated, but not as DNA. During transcription, that segment gets replicated as RNA, or ribonucleic acid. Ribonucleic acid is similar to DNA. It still uses a genetic code. It still uses adenine, guanine, cytosine. But instead of T, instead of thymine, it uses uracil, which is a different amino acid that becomes important during replication. So for now, just remember, during translation, you'll see only U's. During transcription, you'll see a T on the DNA, but a U in any RNA segments. And during this process, there are several other genes that might come into play. The transcription process can be promoted by regulatory genes, genes that say, hey, you need to make this now, and those regulatory genes will turn off at different points during development. That way you don't have basically the body continuing to go through development all the time. Bad things happen then. You'll also have structural genes, genes that code for proteins that actually make up physical body. So we've got those two different kinds. And you may have heard that we only use 90% of our DNA, or 95% of our DNA. And that is true from a certain viewpoint. That 5% of our DNA that we actually use, that is our structural DNA. That's the DNA that's coding, that's making proteins. We used to call all of the other DNA, about 95% or more, junk DNA. Junk is not a scientific term. It is not telling you that that DNA is useless. Instead, it's telling you that that DNA serves a function that's not structural. And for a long time, we didn't know what it was, but we eventually were able to determine that many of these regulatory genes, genes that tell other genes to turn on, turn off, make more, make less, those are coded for in that junk DNA. So these regulatory genes, they are the on-off switches for other genes. On-off may not be quite the right term. It may really be better to think of them as a dimmer switch, a spectrum of fully on to fully off, and everywhere in between. Some examples of things that can happen if that goes awry are things like Marfan syndrome, which scholars think Abraham Lincoln may have had, and it's something we see a lot in people who are very tall and very lanky. Paleontological evidence demonstrates that modern birds evolved from feathered dinosaurs. We can take the DNA of a bird, change the way those regulatory genes are turning on and turning off, and get something that is very similar to a dinosaur. Something with teeth, something with a long tail, something that has scales over more of its body. And we actually have found chicken with these kinds of teeth that are similar to dinosaurs, which is amazing. If that is of interest to you, I strongly suggest reading the book How to Build a Dinosaur. It takes you through that process, kind of reversing these changes over time. We also see something similar with human hair. You have just as many genes to make hair as a chimpanzee. However, the regulatory genes, the genes that say when to turn on and turn off, are different in humans and chimps. So you get different outcomes. You get a very hairy chimp or a slightly less hairy human. Another example of this is lactose intolerance. Lactose intolerance is actually the default state for humans, for all mammals. No mammal is really supposed, quote unquote, to be able to digest lactose as an adult. That's intended for offspring, not for adults. But lactase persistence, the ability to digest lactose as an adult, that is a regulatory gene difference. There's also these homeobox or HOX genes, the homeotic genes. They are kind of master regulatory genes. They are extremely influential at different phases in fetal development. So in the womb, inside, just after fertilization, before birth. Many of these HOX genes are related to positions of different body parts. We've been able to, in fruit flies, for instance, change some of those HOX genes and essentially grow feet where antennae were supposed to be. We've similarly been able to take several different HOX genes together to change the way that some animals are developing during lab experiments. Interestingly, these Hox genes are very similar across species, not necessarily from a fruit fly to a human, but within vertebrates, so from a mouse to goose to python to human. Those Hox genes are very similar and regulate the same kinds of um, things in the development. In this case, the development of the vertebral column or the vertebra. So that brings us to polymorphisms. Polymorphisms are differences in genes, differences in these traits, that we can actually see and measure. They don't necessarily have much of a difference in function. For instance, the human blood type group, AB, AB, or O, that human blood type group is an example of polymorphisms. Um, Many different genes that have similar roles. What protein is on the outside of your blood? There is no such thing as an AB allele. A and B are co-dominant, so both are expressed in someone with AB blood. Because of that, you know their genotype. You know that they have to have the gene for both A blood and B blood. Similarly, if you have someone with type O blood, you know that they can't have either A or B. They have to only have the O allele. And this can result in things like blood transfusions being successful or not. Um, can be very important. However, for an individual, there's not a significant difference in life expectancy or anything with these polymorphisms. So it's not something like a Hox gene where you suddenly start growing an arm where an eye should be. Instead, it's just a difference in the way the body functions. Um, so here are some things for you to ponder. The answers are in the slide material, but if you're able to answer those questions right away, you're probably in pretty good shape at this point. When we get to some other Mendel laws, the laws of segregation and codominance, the law of segregation, remember, says that each of these traits are inherited independently. And that is true as long as, again, they are on different chromosomes. Technically, it is also possible to see this play out if they're on the same chromosome, if they're very far apart, because of that recombination and that crossing over and that transfer of genetic material. If they're far enough apart on a single chromosome, likely you won't be able to tell the difference because that crossing over happens so frequently. Codominance, on the other hand, codominance would be something where both traits are expressed. So in our blood type example, A and B are codominant. You get AB blood. If there was incomplete dominance, then you get a blending of these traits. So you wouldn't get AB blood, you'd get something in the middle. In the case of flowers, you would, if it's codominant, you would get white and red petals on the flower, or white and red segments. But a true incomplete dominance or blending would result in a pink flower. Many traits are not governed by a single gene. Many are polygenic. They have multiple genes that affect their expression. Things like height, skin color, those are not single genes. There's not a tall gene and a short gene in humans. There is in pea plants, but not in humans. Because of that, there's a whole range of heights. You can be anywhere from very short to very tall. Similarly, with skin color, you can be anywhere from very dark to very light. It just depends on how many of these different genes you have. Many traits are also pleiotropic. So in pleiotropy, you have a single gene that influences other genes and other phenotypes. For instance, the sickle cell gene. The sickling trait is a single gene that is inherited in the normal autosomal fashion, so that nice Punnett square works out great for it. But it has effects on other genes. It affects the ability to carry oxygen throughout the body. It affects the ability of blood cells to make it through blood vessels. There's also traits that are the product of genes and the environment. Ultimately, changes in the genetic code that don't make it something that the organism is more or less likely to survive on, they don't get selected for or against, they're just kind of there. Specifically, the environment can play a role in things like epigenetics, which we will have a whole video on shortly. Keep in mind when we're talking about epigenetics, though, that'll be things like nutrition being tied to adult height, where height is genetically controlled, but there's also this environmental factor where you must have the proper nutrition in order to reach your maximum height. Epigenetics, quick precursor to the entire video on epigenetics. Epigenetics are chemical changes in the genome, but they are not chemical changes in the DNA sequence. Instead, epigenetic effects, epi meaning on top, gene meaning gene, so on top of the gene, they're molecules that come in and attach to the DNA or to the chromosome that make it more likely or less likely that that chromosome will be read and interpreted by the body. It can be a result of environmental changes, things like methylation, where methyl, which is similar to ethyl or ethanol, but methylation will occur where it'll come in, attach to the DNA, and make it harder for the DNA to be expressed. Things like temperature changes, environmental chemicals, environmental toxins, disease, nutrition, all of those can play a role in epigenetics. Again, whole film on that. That film is there. Book report, keep in mind that that is still on the list of things to get done. And we are not doing the Model United Nations, so I will go in and delete that slide.